Hello and welcome to this Pharma On podcast, part of our DMPK Insights podcast series. My name is Tim Smith and I am an Executive Director of BD at Pharma On with a specialism in our ADME and PK capability. Today, I'm delighted to be joined by Professor Ian Wilson. Ian trained as a biochemist at the University of Manchester Institute of Science and Technology, UMIST, and following this, with a PhD in the Chemistry Department at Keele University, on a gas chromatographic analysis of ecdysteroids, which are insect molting hormones for those like me who were unaware. Following a short period of postdoctoral research on HPLC-based methods for the analysis of penicillamine at University College in London, he joined Pharma, where he then embarked on a career in drug metabolism and bioanalysis in the pharmaceutical industry spanning over 30 years, beginning in 1979 at Hoechst, then ICI, Zeneca and AstraZeneca. On leaving AstraZeneca, he joined Imperial College in London in 2012, where he is currently a visiting professor of drug metabolism and molecular toxicology. More recently, he has also been appointed as a visiting professor at Liverpool University. As a biochemist turned analytical chemist, he tries to use advanced analytical methods, including those based on UHPLC and ion mobility separations, to better characterize and understand biological systems. Currently, these efforts are focused on high-resolution, high-throughput methods that can be applied to problems in metabolic phenotyping, so metabonomics and metabolomics, drug metabolism, biochemical toxicology, systems biology, and the microbiome. He's the author or co-author of over 600 papers, reviews, or book chapters, and has received awards in separation and analytical science from the Royal Society of Chemistry and the Chromatographic Society, amongst others. From my own very personal view, I was lucky to spend 12 years working at AstraZeneca in DMPK, overlapping with Ian's tenure. What Ian is far too modest to mention in his bio are the impact and learnings he imparted on then relatively young scientists like myself at the time, finding their way in drug metabolism. So it's very much my pleasure to be speaking with you today. Welcome, Ian.
Nice to be here, Tim.
So we could have picked from a wide variety of DMPK areas that you have been involved in over your career, but I particularly wanted to focus on your work around drug-induced liver injury, taking particular aim at the toxicity linked to carboxylic acid-based drugs. Historically, acyl glucuronide metabolites have drawn much attention and fire, and perhaps we can explore your thoughts on the history of acyl glucuronides as possible toxic reactive metabolites.
Well, that would be fun to do, I think, because I've had a 40-year journey with these things, and one of the revelations has happened in the last 20 months or so, possibly thanks to having time to think because of COVID, has been that for 40 years I've been barking up the wrong tree. So many carboxylic acid-based drugs, especially the NSAIDs, are associated with toxicity. And the toxicity has often been associated with reactive metabolites because as drug metabolists, we look out for reactive metabolites. And when we see them, we're not happy. Now, the acyl glucuronides that are produced when glucuronic acid attaches itself to the carboxylic acid of the drug are interesting because they can, although they're biosynthetically formed as the beta-1-acyl glucuronide, they can transacylate, which means an internal rearrangement. So they can go from the 1 to the 2 to the 3 to the 4. And then having got to the 4, they can go back to the 3 and the 2, though technically speaking, they cannot actually easily go back to the 1. In fact, at one time, it was believed that they couldn't go back to the 1-acyl glucuronide. The opinions changed, but thanks to better analytical methods. But that was the thing. Now, if one of these transacylating glucuronides happens to be near a protein, and there are a fair number of proteins in the cell within the plasma, they can transacylate onto things like lysine groups. Now, the theory goes is, how do you make an immunoassay? Well, you stick a drug onto a protein, put it into circulation, and suddenly you've got an immune reaction. And when these NSAIDs turned out to be toxic, it was obvious to the chemists who synthesized them that it couldn't possibly be the drug that was causing the toxicity. It had to be a metabolite. And there we have a reactive metabolite, generally produced in large amounts. So, Tim, that was why it was target. And I met my first acyl glucuronide. It was a Hoechst drug called isoxapac in 1981. So that's where I got the bug. And actually, it was as an analytical chemist that I found it, because HPLC was then a relatively new technique in the lab. Yeah, it was that long ago. And we knew that a glucuride was being formed, and that was the only metabolite. But when we did the HPLC, because up until then, the drug had been analyzed by gas chromatography, which was the technique of drug analysis in those days. And that involved hydrolyzing glucuronides first and then derivatizing them. Well, when we found that we could do direct analysis of urine using HPLC without having to hydrolyze the glucuronic acid or the glucuronide, we thought, well, this is great. We can now analyze both parent drug and glucuronide excreted in the urine, single assay, no messing around with sample preparation. The only problem was we didn't see one acyl glucuronide peak or one glucuronide peak. We saw half a dozen. And as I say, that led to a long period of looking at the analytical methodology because it was just great fun. And secondly, we started reading literature. John Caldwell at the time, very eminent drug metabolist, had also discovered acyl glucuronides. It became a very hot topic. And it was obvious the drugs that had the fastest transacylating, i.e. the most reactive acyl glucuronides, were also the most toxic. So it was definitely a case of quadrat demonstratum. It must be them. Trouble was, it's been a very difficult hypothesis to prove because the wretched things are unstable. So you can't dose them to show that they're toxic because they hydrolyze fairly quickly back in biological media to the aglycone. So trying to tease apart which is the toxin and which isn't is tricky. And then to muddy the waters a little further, other scientists said, well, hang on. These are carboxylic acid drugs. Metabolism is being hijacked by them, not just to make acyl glucuronides, but they also make coacyl A esters. And we know this because you see taurine conjugates and other amino acid conjugates, glycine conjugates in particular. So they have to go through CoA to make that. Now you don't see the CoA because it's used quickly, and also CoA conjugates are wonderfully unstable. They're reactive. And guess what? If you dose radio-labeled drugs into a system where you're making CoA conjugates with your carboxylic acids, you get stonkingly high quantities of covalent binding, up to 10 times what you see in terms of covalent binding compared to carboxylic acid. So now we have two candidates for toxicity, and that's where it gets really interesting. So my colleague Lars Whitehoff, with whom I've recently written a perspective on the topic, was an acyl-CoA man. I was a carboxylic acid goes to acyl glucuronides man. So we made a perfectly complementary team. And so you had the drug metabolism world divided roughly into two unequally sized groups, those that believed that the acyl glucuronides were the toxin and were looking for proof of that, and those who were looking for the CoA conjugates as being reactive metabolite and looking for proof of that. And probably, Tim, therein lies the problem, because after approximately 40 years of looking at this, there was still no conclusive proof that the carboxylic acid containing drugs were toxic through either route, really. But certainly it was looking a bit dodgy from the acyl glucuronides. Now, that said, whilst no one could show causation, there was this definite correlation. So the acetic acid type acyl glucuronide formers, like diclofenac is a particularly good example, compared to things like ibuprofen. Both form acyl glucuronides. One's a lot more problematic than the other. I mean, ibuprofen is a really very safe drug. Now, what people forget when they look at ibuprofen is that there's an earlier analog of ibuprofen that Boots actually wanted to market, well, did bring to market first, called ibufenac. Now, that was a straight acetic acid, and it was withdrawn within a couple of months of launch, really. Ibuprofen was just the propionic acid. It was an alpha methyl analog. The RNS forms. The ibufenac transacylates really rapidly, half-lives of between an hour and hour and a half, depending on who's doing the analysis. Whereas ibuprofen has got a considerably longer half-life. Although, interestingly enough, the R and the S forms transacylate at different rates. So when you do it on the racemic drug, you make the racemic glucuronide, you're getting a hybrid half-life. Now, I want to keep on the ibuprofen versus ibufenac story because it's one of the few cases where we have both the acetic acid and the phenylpropionic acid, and the propionic acid. And the difference is stark and clear. And it's at the distance of 40 years now, it's surprising to me that the chemists never really got the message. Or maybe those at Squibb did, because when they produced naproxen, they produced S-naproxen rather than RS. And that proves to be even cleverer, because one of the stunning things that happens, and you have to marvel at the biochemical insight that went into doing this. If you dose R and S ibuprofen, and you look at the ibuprofen acyl glucuronide that's excreted in the urine, there's a very heavy conversion from the R to the S. You only see the S in there. Now, the only way you can do that is you go through a CoA intermediate, which is planar, which then hydrolyzes, and it only goes in one direction. You don't seem to make very much R from S, but you make a lot of S from R. So your body is actually playing the trick on the drug metabolist by converting potentially toxic R enantiomer with a fast transacylation rate into a slow transacylation rate. So, if drug metabolism is a game of snakes and ladders, making the RS, the racemate, is a definite ladder, because it all gets converted to the active S. And isn't that beautiful? Because it is the S that's active and the R isn't particularly.
And it's via the CoA intermediate.
And it's via the CoA intermediate. Now, if CoA is a route to toxicity, why is ibuprofen not toxic?
Absolutely.
But as we said, there is a correlation between the half-life of the acyl glucuronide and toxicity, even though I finally no longer believe that the acyl glucuronides are primary toxins, or at least not through reactive metabolism, or at least not through the generalized reactive metabolism you see. I mean, if you look for, reactive protein adducts in plasma, and you look for diclofenac, Kevin Park and his group did a experiment where they looked at, it was only 6 patients, but every single one of those patients had adducts to their human serum albumin direct from their circulation, and not one of them seemed to be showing any adverse signs. So tricky. But the acyl CoAs, okay, much more reactive, but still, not a smoking gun, probably. So if you like, if you're developing a drug that's a carboxylic acid, or one of its major metabolites is a carboxylic acid, and you see an acyl glucuronide, it's not the acyl glucuronide you should be worried about, because it's probably not the toxin. But if it has a fast half-life, you should begin to get worried, because what the acyl glucuronide is, effectively, it's the canary in the mine. If you see one and it falls over because it's got a fast half-life, it probably means that if you have a high-dose drug with a significant throughput to the acyl glucuronide, that it has a good chance of being toxic. But it's not the acyl glucuronide that you see, it's the coacyl A conjugate that you don't see. Because what I now believe, and I think what Lars and I are pushing in this perspective that we've written, is that the coacyl A is a route into all sorts of other types of metabolism. So if you look at what happens to coacyl A metabolites, well, they get converted into amino acid conjugates. Right. What other things get converted into coacyl A conjugates? Because remember, what we've got here is a drug hacking its way into a pathway that wasn't designed for it, I mean, 4.5 billion years of evolution didn't go into getting ready for naproxen, right? It's
No.
nonsense. So what's happening is that the drug is a substrate for an enzyme that's moderately promiscuous. And so, bile acids are an obvious thing that are formed by, tyrocolic acid. The clue's in the name. It's a taurine conjugate of cholic acid. So if you look at the metabolism of many non-steroidals, you will see in rodents, especially in mice, but also in dogs as well. I'm aware dogs aren't rodents, but I'm wandering over my species here. You'll see taurine conjugates. If you look at them, you probably also see glycine conjugates, and you'll see acyl glucuronides as well. So you get a mixture. Humans are not necessarily so good at making taurine conjugates of some of these things. So, but in rat and mouse hepatocytes, you obviously have another way of seeing the flow through the CoA pathway. So you can't see the CoA itself, but you can see the downstream metabolites. Now, for a particular drug of interest of mine, a drug called phenoclosic acid, which was initially developed by ICI, about the same time as diclofenac was being developed as well. And it was a fantastic drug if you're a chemist, because it had all those things we don't get anymore. It was water soluble, very water soluble. It was very effective. So 200 milligrams of phenoclosic acid could replace 4 grams of aspirin per day, which if you're an arthritic is a really good thing to do, because we all know what aspirin does to stomachs. And it was progressing really very, very nicely until they decided that in order to get all day cover, they needed to put dose up to 400 milligrams. And they put it up to 400 milligrams because they didn't want to give 2 divided doses, because patients couldn't cope with doing 2 divided doses, even if they were taking 4 grams of aspirin a day. And at that point, something, and I forgive me for not having the exact numbers, but out of 15 volunteers in Aberdeen, 4 went yellow from having liver problems. Now, that's not idiosyncratic. I can never remember what the opposite of idiosyncratic is, but let's call it syncratic.
It's very much syncratic,
yeah. This is not an accident. And if you look at the metabolism of that compound in liver humanized mice, you find a whole menagerie of unusual conjugates. You also see things like side chain lengthening and side chain shortening. Side chain shortening, for Pete's sake. Now, phenoclosic acid is very similar to diclofenac in that it's an acetic acid, right? So it forms acyl glucuronides really well, and that was the major metabolite in humans. But we have all these other things going on that clearly implicate CoA, and a vast number of things that implicate different routes. So side chain, the formation of acyl carnitine conjugates. How do you get fats into mitochondria? Well, you make carnitine conjugates of them, which are transported in. I mean, that's one of the ways you get things into mitochondria. And here we have a potential mitochondrial toxin. You get taurine conjugates. Okay. Taurine conjugates are transported if they're a bile acid. If we take as a basic thought that, these carboxylic acids are like cholic acid, hacking into the bile acid pathways, they're going to be transported out in the bile. Okay. And the glucuronides too. Now, let us start thinking about transporters as a toxicity rather than reactive metabolites. So what do you think about that, Tim?
Well, it sounds to me like we've just created a very efficient, or they've hijacked a very efficient carrier mechanism to get them into the place of interest to exert the toxicity.
Indeed so. But because we've been fixated on it's the acyl glucuride reactive metabolite that's the toxin, or the acyl CoA itself as a reactive metabolite, the toxin, we haven't bothered to look at the downstream consequences of either of these things. Now, as we've discussed in the past, the manufacture of the amino acid conjugates from the parent drug is sort of trivial O-level or GCSE or whatever it is these days chemistry. When was the last time anybody ever asked you to look at the transport and toxicity of a metabolite? Especially one that is a conjugate, when everybody knows that conjugates are benign. Well, not
apparently
if they're
so.
No, they're not going to be benign if they're sitting in the wrong place. Now, it is just possible. I diss the idea that the acyl glucurides could be a toxin based on reactivity, but if they react with the transporter that's trying to push them into the bile, that actually changes the story a little. So they may contribute in that way. And it could well be that I just spent too long in the sun and I've gone slightly mad. And it is in fact acyl glucurides that are toxic. But show me the killer experiment that proves the hypothesis. So I think we need to get now to the science. Let's talk about science philosophically. If we do science properly, you develop a hypothesis and you devise experiments to prove it. And if those experiments succeed in proving it, if you're a good scientist, you say, what have I done wrong? What would disprove what I've just proved? And if you try very hard to come up with experiments to disprove things, very hard, and you can't disprove them, then there's a chance your hypothesis is tenable. If you say, well, reactive metabolites, paracetamol, toxic, we've got reactivity here with acyl glucuronides, not in the same league as paracetamol, but the toxicity doesn't develop as quickly. Toxic. We don't have to think anymore. Or you do the same thing with CoA. I'm suggesting that probably if you're just doing drug development, it doesn't matter because you can do the simple equation. What's the dose?
Yeah.
What are the pathways? It's an acyl glucuronide. It's a gram a day, and this is made up. This comes up to 60 or 70% of the metabolism in humans, and it's for headaches. We probably have a problem here.
Yeah.
It's given at 1 milligram per day. It's the acyl glucuride is 10% of the total metabolism that we can see, and it's given for 5 days. We don't have a problem.
Let's proceed.
You don't have. You can do the risk assessment without knowing the effect. But if you actually are trying to get a drug through where the therapeutic index isn't that great, but it is quite important, then you might want to think a bit more. I mean, with Ziglifan, PAC 875, which is a recent failure, look at the structure sometime. It's effectively an acetic acid on an aromatic ring. It's got what looks to me like an almost taurine analog at the other end. It's a large molecule. It looks like a bile acid. And it does all sorts of things. The drug itself interferes with transporters, which suggests that it is a transporter substrate. You know what I mean? It has the unfortunate habit in the dog of crystallizing out in the bile, which might explain liver toxicity in the dog at sort of dose. But I mean, it failed in humans in late stage 3. Now, they knew that there was an acyl glucuronide formed early. It's a reasonably high dose drug. That should have sent all sorts of alarm bells ringing. Now, what I'm saying is, okay, those are the alarm bells. What's the mechanism? And the only way we're going to find out what the mechanisms are by looking more deeply. And I think we need to look down some of these lipidic pathways that you get in the mitochondria, and also start looking at things like how are taurine conjugates transported. Let's make the taurine conjugate of diclofenac. and let's see whether that actually inhibits bile acid transport. Yeah, that's such a trivial experiment to do. As I say, if I was a competent chemist, and I've never claimed to be competent, I'm certainly much less competent now. This is something you could stew up in your bathroom or in your kitchen, because the chemistry is quite simple. You've got assays, although, you know, one of the things we now routinely do is measure transporter inhibition.
Yeah.
So if you've got metabolites that are benign because they're conjugates, I think any sensible pharma or any sensible DMPK person in pharma would say to their chemists, please make me these, because I'm kind of worried that they might be inhibitory, and that's something we ought to know, especially if it's a high dose drug. I mean, yeah, again, rule of thumb, depending on the dose, you've got a fairly good idea of, you know, we all know the mantra high dose, or rather what we actually mean is high bioavailability. High dose
Yeah.
is more likely to be toxic than low bioavailability, low dose, or just
Low dose.
low dose. Now, that depends on whether your drug is cyanide or an antibiotic. But, you know, the risk assessment becomes a lot easier if you know what the dose is. Now, of course, one of the problems in drug development is you're never really certain what the dose is going to be. So, you know, there are things that you can do. And one of the things that Lars and I have suggested in our paper is if you've got an acidic group dangling on the end of a chain sticking off an aromatic group, make it an ibuprofen analog, not an ibufenac analog, because if you stick that methyl in, and better still, make it the S-methyl, you will completely block a whole set of pathways that are potentially toxic. You won't block making necessarily ester glucuronides, but I'm telling you, ester glucuronides are not something you really need to worry that much about. What the ester glucuronide is telling you is that you have a reactive carboxylic acid. It's not a toxophore on its own, but it's telling you there is a potential toxophore there. And you know that you'll slow down transacylation if you have the S form anyway. I mean, it's one of those really wonderful ways of remembering things, because if you've got the R form, the transacylation is rapid. And if you've got the S form, the transacylation
it's
is slow. Yeah, I mean,
even I can remember that.
Yeah, this is simple and very basic stuff. So, you know, if there are any chemists out there listening who would like to do a bit of exploratory work on this, publication guaranteed, they should get in touch with me.
As we've discussed, Ian, it's that whole sort of series of pairs that you can make and test and hopefully show you know that correlation.
Yeah, and again, as we have discussed, the mitochondria are the forgotten organ of metabolism in the cell, or the forgotten organelle. We all worry about mitochondrial toxicity, but we never investigate mitochondrial metabolism, and there's a huge amount of mitochondrial metabolism goes on. I wonder if valproic acid, which is another acidic drug, which I haven't really looked into, but which has got all sorts of problems associated with it.
That has a very, very fast half-life.
Yeah.
For the acyl
has anybody looked at the acyl carnitine of valproic acid? Does it get into the mitochondria? I mean, it looks like a fatty acid to me. Well, you know, there's a whole. Once you get away from the fixation of acyl glucuronides and to an extent CoA and say, okay, what are the downstream consequences of these metabolic pathways? Okay, looks like acyl glucuronide formation is a true attempt at detoxification, but not CoA formation. That's a true attempt to get into a whole load of downstream endogenous metabolism. And we have the tools to investigate that now. Many of these experiments can be broken up into small quanta. You don't have to do everything at once. So testing transporters on a couple of taurine conjugates shouldn't be difficult. making the analogues, the ibuprofen and the ibufenac analogues of some successful and safe and some successful but toxic NSAIDs, or just toxic NSAIDs, would be very interesting. I'd love to see what would happen if somebody made the ibuprofen analog of phenoclosic acid.
Yeah.
Which, by the way, if you look at it as a drug metabolist, is a wonderful compound because every part of it looks dangerous. Yeah, got aromatic ring with a chlorine on it. Oh yeah, okay, it's got to get metabolized there. It's got a thiophene ring. Well, don't talk to me about thiophene rings. And then it's got this acetic acid. So all in all, it's a disaster area. Everything was made a lot easier by the FDA declaring that acyl glucuronides were toxic.
Yeah.
Yeah. So that gave an extra impetus to looking at acyl glucuronides. But as I say, it is a bit like we always forget, because it's easy to forget, that correlation and causation are different. And
two things.
Yeah. But you can use the correlation to help you make decisions. You don't have to have it's arguable that you can get to the moon using Newtonian mechanics. You don't need Einstein, but Einstein took Newtonian stuff and moved it on to the next level.
Yeah.
Yeah. So I think the acyl glucuronides and CoA stuff are a bit like that. You can link them to things, and they go in lockstep, but it's what happens afterwards that's really important.
Yeah.
But people who are interested in this need to look at the literature again themselves and re-examine it. One of the nice things is now we've got computational methods that are good enough that you can synthesize the glucuronide in your computer and then predict its half-life. Yeah, in your computer. So you don't actually have to make the damn molecule. You can just imagine it. And the correlations with real and computational results seem to be pretty good. So they may be out by a half hour or so, but they're still good enough. And I know at a recent DNDG meeting in Switzerland, somebody there was presenting on how they were using computational chemistry to predict half-life of acyl glucuronides. So this is a live topic, and it needs to stay live. But people need to understand, I think, the potential limitations of the arguments that are being used.
Absolutely. Thank you, Ian, for such an illuminating discussion, and thank you very much for your time. Perhaps carboxylic acid functionality can now be viewed with a little discretion. We've moved a long way from the historically held beliefs that these groups are to be avoided. Clearly, there are mitigation and testing strategies that can be employed that will provide more confidence to discovery and development teams looking to move forwards in this area. Thank you all for listening to this episode of Pharmaron's DMPK Insights series. We would like to remind you that our DMPK webinar series is also available on demand, covering a variety of key questions related to DMPK science in drug discovery and development. Stay tuned for more podcasts in our Pharmaron DMPK Insights series. Thank you, and bye for now.